so our last featured speaker this morning is Jared Homola from Wisconsin Cooperative Fisheries Research Unit. And he's going to be talking about some genetics work. We're coming back to Wisconsin, but I'm hoping there may be some results even from a few places around the country. So I will turn it over to Jared.
Great. Thank you, Sarah.
Yep.
I appreciate the introduction and the invitation to come speak with at the symposium today. So I'm going to walk through some different genetic considerations that folks tend to want to keep in mind when they're trying to come up with brook trout propagation and stocking programs. As Sarah mentioned, I'm with the Wisconsin Cooperative Fishery Research Unit, and I also lead the Molecular Conservation Genetics Lab here at UWSP. So most of my talk will be focused on Wisconsin, but here in Wisconsin, brook trout stocking has a really long history that's been evolving, especially more recently. So brook trout have been getting stocked in Wisconsin for well over 100 years, and always in the hundreds of thousands of fish. For a long time, there was a domesticated strain, the Osceola strain, that was most widely used, and then they eventually moved on to the St. Croix Falls strain, which originally came from New Hampshire out east, but again was a domesticated strain of brook trout. And then more recently, I guess coming up on nearly 30 years ago now, biologists have been more and more looking toward wild broodstock sources that can be used on a rotation and used more locally in different parts of the state. So those are the types of considerations that my talk today is going to focus on, is choosing a broodstock source in order to preserve the genetic integrity the best we can for these native populations. So there's a few considerations offhand that I want to acknowledge. There's a desire to be able to go to streams and populations with high abundance if you're trying to select a wild broodstock source. There's been realized concerns in the past about declining abundance if we keep going back to the same stream over and over again to collect eggs and mills. Of course, you want the individuals in the population to be healthy. We don't want to be moving diseased or disease-susceptible individuals around the landscape. They have to have an accessible location, so field crews have to be able to get in and safely work with the fish. It's useful to be fairly proximal to a hatchery. So again, the crew is going out there to monitor the ripeness of the fish, and the spawning activity going on can get there readily and consistently. And then there's a need to consider appropriate genetic characteristics. And I go through this list to be sure that I'm not being dismissive of any of the first 4 points here. I'm just going to focus, though, based on my expertise, on the last point here, that we're choosing appropriate genetic characteristics for our broodstock sources. And there's 3 aspects I'm going to talk about today. So one is looking for adequate genetic diversity within an individual population. So this is a population level metric. We also want to be sure that we have, as best we can, high genetic relatedness to the recipient populations that are going to receive the offspring from this broodstock source. And we want to look for low evidence of domestic brook trout introgression from past, and in some cases still ongoing, practices of stocking out some domesticated fish that we don't want to be sending on those genetic variants to other places if we can help it. So for the rest of the talk, I'm going to go through each one of these 3 considerations, and I've set this up to first provide kind of a schematic overview of why that consideration is important. And then I'll present some of the best available research out there for how that theory translates into the real world and out into wild populations. And then provide a snapshot of how those factors are looking here in Wisconsin. So first, looking at within population genetic diversity. So consider 2 different populations that are perhaps being evaluated for their suitability to be a broodstock source. On the left, we have a high diversity population, and I'm just using color here as a surrogate for genetic diversity. On the right, a low diversity population. So the first way that this the first reason that this factor is important is fairly intuitive. Say there is some threat that comes in to our brook trout fisheries in Wisconsin, and the only individuals that are able to deal with that challenge, so maybe it's a disease, maybe it's water warming, or say it's the orange individuals there in the high diversity population. So what's going to happen is you're going to lose everybody that's not adapted to that. And because some individuals were able to hang on to the high diversity population, they have a chance to to grow back again through adaptation to this new environment or this challenge. Whereas low diversity population, perhaps it got to such low abundance that it blinked out entirely. Of course, it's the same reason that connectivity is important between these different populations, because so perhaps the high diversity individuals or that population can go recolonize the low diversity area. And this idea of is often referred to as the adaptive capacity of the population, which is proportional to the genetic diversity. And there's been a lot of work that's been done to see if this theoretical connection holds out in the wild. And indeed, this recent study by DeWoody and colleagues was a meta-analysis that looked at genetic diversity on the y-axis. So this is HE, expected heterozygosity. Just think of it as one of many measures of genetic diversity, and how that varied across a lot of different species, and grouping it by taxa here, where the white column on the left are species that are threatened, and then on the right are threatened or endangered, on the right are not threatened or endangered, so stable species. And regardless of whether you're looking at mammals, birds, reptiles, amphibians, fish, or lumping everything together, there's this really consistent pattern that the more stable populations, more stable species have greater levels of genetic diversity. You might be thinking that's kind of a chicken and the egg situation, right? If something is starting to decline, it's probably going to lose some diversity because there's just not as many individuals out there to hold diversity. And that's true. So what you end up seeing sometimes is what can be called an extinction vortex, where you get this feedback loop where declining genetic diversity is met with declining number of individuals, and these two things feed back on one another to make the situation worse and worse. And that often will lead to extinction or extirpation at the population level. So that's one reason that this genetic diversity has been seen to matter. But that's more forward looking, and we want to keep our brook trout populations out there on the landscape, but also there's evidence that just individual level heterozygosity, so just whether an individual lives or dies, is also reflected in genetic diversity measures. So this was a great example in desert tortoises from a few years ago, where these tortoises were translocated into a new environment, and how genetically diverse an individual was ended up being significantly predictive of whether or not it would survive that relocation. So these two figures on the screen right now are kind of part and parcel to each other, where on the right there, whether or not something lives or dies is related to how diverse it is, but then that really extrapolates across species and populations. So how are we doing with this in Wisconsin and the streams that I study? So this and many of the results I'm going to have, you'll see in the bottom left there, Tometz et al. I want to acknowledge the graduate student on this project that did these analyses I'm going to be sharing with you today, the grad student in my lab. So what Andy found here across all these populations that he surveyed in Wisconsin, each shown by a colored dot there on the map, is really a range of expected heterozygosity values. So just like that figure on the left from the last slide, this is the exact same metric that we calculate for our brook trout populations here. And you see a speckled landscape here. And there's a general trend of more genetic diversity as you get further to the north in the state. but there's certainly pockets of higher levels of diversity, like down in the Driftless region in the Southwest. You can see some of our lowest heterozygosity values, but also some of the highest as well. So there's really a lot of localized variation there. A second metric that some folks might hear about sometimes is effective population size. So this is a metric that generally captures the number of individuals that are spawning successfully and passing their genetic material on to the next generation. We want to have high effective population sizes for broodstock sources. Here, not as rosy of a picture down in the Driftless, but as you move to the north, again, Wisconsin, you get more diversity, but the scale is kind of skewed because there are a few very high effective population size. So really, if we were to truncate the scale, you would see more variation in the Driftless, where again, there are populations that hold more diversity there. Second consideration, this is that among population genetic divergence. So consider a scenario here where you have one population that perhaps is more warm water adapted, and a second that is more cool water adapted. If you were to take individuals from the cool water population, use those as a broodstock source to stock out in the warm water, and quite intuitively, those fish are eventually going to reproduce, and what you get is this shift in adaptiveness where maybe those warm water adaptive traits are getting diluted. So you can shift kind of the genetic adaptive landscape of a population through the stocking. And this process is termed outbreeding depression, where you're taking two things that have different adaptations, crossing them, and you end up with something that's more moderate that may not really be adapted to any specific situation. A really elegant study, I think, that shows this was in Minnesota populations of slimy sculpin. And in this study, and in these figures, we have G and B are the two original populations that it turns out were quite divergent from each other. When you cross them, excuse me, when you cross them to create an F1 population, you get reductions in these metrics, both growth and weight. If you go on to an F2 population, so you're crossing individuals from F1, it gets more amplified. So you're getting these individuals that are just performing worse because of this cross of divergent lineages. And then in the far right side of these figures, if you take the F1s and back cross them to those original lineages, you start to direct it back into a certain adaptive direction, and you can reverse some of those declines in some cases. So this is, again, just an example of outbreeding depression and the sort of thing that we worry about if we're taking broodstock and stocking them out someplace that is quite divergent from the original broodstock source. Of course, you can't go out and get genetic data and understand adaptation across every population and every recipient water body, every broodstock source. It's a daunting and really untenable task. So what we do instead is use geography as a surrogate for this. So we can capture a lot of this variation if we just stick things, make sure our recipient water bodies are close to where the broodstock are from. And this is how Wisconsin is dealing with this question. And this map was some genetic management units that were formed based on the genetic data we have on multiple species. So if we don't have detailed information on a given species, this is kind of the default of where we should draw the lines of taking broodstock individuals and where their offspring go. But what does it look like for a brook trout again? So in this case, again, more of Andy Tometz's thesis work, here the colors represent different genetic lineages. There's 9 different lineages shown here. It's just one configuration of this model that explains the data well. Each of the pie charts are a population that was sampled. And the coloration is the proportion of individuals that were most closely related to one of those 9 lineages. So you can see here is a lot of variation out there, and that's thought to be caused by 2 different factors. One is the extensive stocking history. So our native genetic structure has been skewed quite a bit. And secondly, some of those low effective population sizes lead to genetic drift, which is just random variation that can come into this genetic structuring. And it can make individuals become less closely related or make populations less closely related just by chance alone. So with our brook trout, though, there is some geography that comes through, such as in the central part of the state, this green cluster 3 that's pretty prevalent. Up to the north, we have blue that's pretty prevalent. Again, what most folks in this group are interested in down here in the driftless, much more of a scatter shot. There's not a lot of genetic structure down there. A couple neighboring populations shown here at the 7th cluster, which is distinct to anywhere in the state, so potentially some interest there. But by and large, there's just a lot of variation in the lineages that are down there in the southwestern part of the state. Our third consideration, the past influence of domesticated fish stocking. So this is kind of an amplified example of the situation I was just talking about with the second consideration of outbreeding depression. So you consider a native wild population, and I have this gradient down here between a wild and hatchery adaptive, and a domesticated brood stock. So these are fish that have been held in a hatchery for many generations. And through that time, they become adapted to their environment. So they're more apt to eating the food that they're given in a hatchery rather than foraging necessarily. There's been studies that have shown changes in aggressiveness and movement propensities and all sorts of things based on this hatchery adaptation. And the thing, so yes, if we have some individuals move over because we're using that as brood stock, you might shift so that the population that's out there through time becomes less well adapted to the wild setting and the traits they need to have to survive there. Now, one of the most striking things about this is how quickly it can happen. So this is 2 studies on steelhead that both show effects after only 1 generation in the hatchery. So on the left here, this is the y-axis is relative reproductive success. So 1 is indicative of an even amount of reproductive success, so ability to successfully reproduce between individuals that were raised in a hatchery versus those that weren't. So 1 is our benchmark here on this figure on the left. After just 1 generation in captivity, you already have a decline in those hatchery reared individuals' ability to reproduce out in the wild. After 2, you have a substantial decline. And what the authors of this study did is they then looked to other studies for longer generation times in the hatchery, and this curve just continued to go downward. Where if you get out to 10, 12 generations removed from the wild, you have very little reproductive success of those individuals out in the wild. The study on the right here, again, shows just after 1 generation with these steelhead, HH, this is hatchery crossed with hatchery on the left, wild crossed with wild on the right. And again, the wild individuals are outperforming in their reproductive capacity the ones that were reared in a hatchery. And interestingly, in this study as well, I don't have the figure up, but they also tracked the fish's ability to grow. And the hatchery fish grew better in the hatchery than the wild fish after just 1 generation. So again, just another sign of how quickly adaptation can occur to these environments. So our look at this involved doing assignment testing. So this is an example result where we have 2 different lineages shown. Hatchery is representative of the St. Croix Falls domesticated strain. Blue is the native strains. This is kind of a grouping of all what we can best decipher to be native genetic characteristics of the brook trout across Wisconsin. Each one of these boxes is a different population. And I've zoomed in on 2 of the populations here, so Melanchthon Creek on the left and Willow on the right. Each one of these bars represents the probability that a single individual is either of native or hatchery ancestry. So you can see in Melanchthon, everything comes back as being 100% native. We don't have any evidence of these hatchery genetic variants. But in willow, it's the opposite story, where it's much more influenced by the St. Croix Falls strain that's been stocked there in the past. So these are all of the streams that Andy studied for his thesis research, and these are all the ones that were pointed out on the map. I'll put it back on a map in a second. But you can see in the Great Lakes region, so the Great Lakes Huck 2, we have much less stocking influence than we do in the upper Mississippi on the west side of the state. And even there, yeah, I'll go over to the map. So this shows those proportions. Again, driftless, quite a bit of hatchery influence. And importantly, for Andy's study, the populations you see here, we selected those based on having no documented history of stocking. So what we see here means one of two things. Either they were stocked with domesticated fish and we just didn't have the record, or what we think is more likely is it was a neighboring stream that was stocked with domesticated fish that then strayed out of that stocked location and into these study water bodies. So again, we selected populations based on limited evidence of stocking. There was another study just a couple of years ago led by Brad Erdman that asked the same question. And Brad's study was more of a random grab of different brook trout throughout Wisconsin, brook trout populations. And here, so you give a less biased take on evidence of past stocking. So you see a lot of spots in the driftless region that seem to be heavily influenced by these domesticated St. Croix strain fish, but it's certainly not everywhere. There's a plenty of locations that, from this perspective, could be adequate broodstock sources that don't have any history of this. A really neat analysis from that Erdman paper looked at a process called allelic purging. So if these domesticated fish are getting out there, how long do their genetic materials, how long do their alleles stay in that population? And what they found is as you approach 50 years since the last stocking event, what you see is this reduction in the hatchery identity or those hatchery genetic variants that are still in the population. And this is expected because they're expected to be less well adapted. So even though the hatchery fish themselves died out long ago, their progeny are also less successful. And over time, you have this loss of those poorly adapted variants. So it's really an encouraging story that despite seeing a lot of these signals out on the landscape, through time, they do tend to fade. So just in conclusion, to summarize what I've gone over here. So different considerations. First, adequate genetic diversity within populations. Here in Wisconsin, diversity is really variable throughout the state, but tends to be greatest in the north. We want high genetic relatedness between our broodstock and recipient populations. And there's spatial structure in some places in Wisconsin, but geography isn't necessarily the best surrogate for this relatedness. And elsewhere, where there's not clear spatial genetic structure, the default ought to remain keeping broodstock and recipient populations close together, which may allow that spatial genetic structure to reform. Also, there's we want low evidence of domestic broodstock or brook trout introgression, and more than half of the populations that we looked at across our study and the Erdman one had no evidence of stocking history. So while this is something to keep an eye on, it's certainly not widespread throughout all of our populations. And an encouraging point is that this allelic purging tends to help erase those domesticated genetics as time goes on. If there's time, I'm happy to take any questions or discuss this. But again, just want to acknowledge my graduate student, Andy Chmetz, there in the upper right, who is the student that generated these data and did a lot of the analyses for what I talked about today. Thanks, everyone.
Thanks so much, Jared. That was really interesting.